The research in this study was conducted by my PhD student, McLeod, who focused on investigating fumigants and semi-selectives for managing apple replant disease in South Africa. The research was conducted in order to improve the management of ARD because growers sometimes report that fumigation with chlorpyrifene 1,3-dichloropiprine is not effective. This could be due to two reasons. Either the fumigant was not applied correctly or the nursery material that was used contained ARD pathogens. In a previous study, we showed that it is problematic that apple nursery trees can contain ARD pathogens, especially lesion nematode often occurs on these materials. So in an attempt to address this problem, we evaluated semi-selective chemicals, specifically to address the problem of nursery materials being contaminated with a pathogen. And then we looked at different formulations of chlorpic because there is not much information which formulation is the best to use. The chemicals that were used in the semi-selective mix were aimed at the different groups of replant pathogens, which include nematodes, oomycetes, and fungi. The mix also included imidacloprid, which although woolly apple aphid is not part of the replant organisms, it is often associated with nursery trees. So since we tried to address the problem of poor nursery material, we also included imidacloprid within the mix along with namifos to manage the lesion nematode and phosphonates and metalaxyl or mefenoxam to control Pythium and Phytophthora species. The mix did not include something specific for the fungi involved because most fungicides are not systemic and we therefore could not identify an appropriate candidate. 3 orchard trials were established, 2 in the Koa Bokkeveld, namely WZB and KBC, and 1 in the Grabouw region, GRA. The treatments included the 2 different formulations of piclor, which included one that contained 33% chlorpyrifene, whereas the other one had a higher concentration of 57%. Then we had a treatment consisting only of the semi-selectives, the fumigation treatment combined with semi-selectives, and then in 2 of the trials, we still had methyl bromide available. So we included this as the gold standard that previous years used. And then 1 of the trials, we included the higher content chlorpyrifene along with the semi-selectives. The trials were evaluated by determining the increase in trunk diameter over time as well as yield. The increase in trunk diameter measurement showed that all of the treatments were significantly better than the untreated control from the first year onwards, and also that they did not differ significantly from each other. The only exception was the semi-selective treatment, which is the green line, which at the KBC trial only caught up to the other treatments in year 3. The graph shows the percentage increase in yield relative to the untreated control. In 2 of the trials GRI and KBC, a good response was seen to fumigation, which is the light blue and green bars. In both trials, the fumigant with the higher chlorpyrifene content, the 75% one, outperformed the PIC 33 fumigant. In the graph, the bars or treatments that have an asterisk above it did not differ significantly from the control when we assessed the yields. The semi-selective treatment used on its own, which is the yellow bar, was only effective in the KBC trial in reducing yield significantly better than the control, but not in the GRI trial or the WZB trial. This is most likely due to differences in the ARD pathogens present at the different trial sites. The WZB trial did not respond to fumigation or to any of the other treatments. As you can see, there was no significant difference in yield between any of the treatments and the untreated control. There was, however, a strong trend that the methyl bromide and the PIC33 plus semi-selectives increase yield at least 60% relative to the control. The most likely reason why treatments were ineffective at this trial is because tree roots from the previous planted orchard was not removed effectively, and the new orchard was established too soon after removal of the previous orchard, being not longer than 1 year. And alternatively, it is possible that the nursery trees were contaminated with ARD pathogens, which can also render fumigation and other treatments ineffective. To conclude, it is recommended to rather use a higher chlorpyrifos content fumigant within the range of 50% rather than those with chlorpyrifos in the range of 30%. Then semi-selective chemicals may have the potential to improve fumigants, especially in situations where all tree roots were not removed effectively, as well as the use of nursery trees contaminated with ARD pathogens. However, we will need to do more work on this to confirm whether this is true. I would like to acknowledge my project team, of which Mako Nyoni is the most important since he did all of the hard work during his PhD study. Also, Becker Wessels and Mark Mazzola for their critical input and suggestions during the course of this study.